Hi, my name is Bailey Turee, and today we're going to be talking about the effects of varroa mite parasitization on honeybee colonies. So first we're going to talk about the varroa mite itself and their origin. So the varroa destructor is the scientific name for what is commonly known as the varroa mite. Varroa mites originally preyed on Apis cerana, but through evolution, Apis cerana were found to have natural means of protection against these parasites. When Apis mellifera were brought over to Asia, people found that this mite was most devastating to this species. Over time, this parasite began to prey on Apis mellifera and spread around the world. The results of this pest are so severe that countries without an infestation take quarantine procedures to prevent one. So here is a timeline of the varroa mite infestation in the United States. So mites were first found on Apis mellifera in Hong Kong and Singapore in 1963. In the early to mid 1970s, varroa mites appeared in various countries in Europe and South America. In 1979, a single mite appeared in Maryland, and in 1987, varroa mite colonies were found in Florida and Wisconsin. And today, all continents except for Australia have a varroa mite infestation. So spotting a varroa mite. On the picture to the left here, you can see this adult varroa mite, and you can see it with a naked eye, and they're commonly found here between the segments on the abdomen. Here, these are two adult female varroa mites on a honeybee pupa. So varroa mite reproduction. Adult female varroa are found on immature or adult honeybees. Male varroa never leave the brood cells, and immature varroa can only be found on capped brood. The adult female undergoes two phases depicted below. So the first phase is the phoretic phase. The adult female feeds on adult bees and are passed around the colony. During this phase, females live on the bees within their segments on their abdomen. The varroa puncture the bees and feed on their hemolymph. Moving on to the reproductive phase, phase 2, females must enter the bee brood cells to lay eggs before bees cap the cell. Once the cell is capped, the mite releases tubules, which allow the mites to breathe while capped in the brood. Once the bee larvae consume the remaining brood, the mite attaches to the larvae and begin feeding. This mite will lay its egg, and if unfertilized, it will turn into a male mite. So the anatomy of a varroa mite. Their natural curvature of their bodies allows them to escape the cleaning practices of the honeybee. The claws allow them to attach to the bee, and they also have a ventral setae that allow them to remain attached. The cuticle of this mite has a similar composition of the bee, which allows them to go unnoticed if attached, as well as protect them from bee aggression. So when discussing the relationship between the varroa destructor and the Apis mellifera, varroa destructors have been found to be transmitted horizontally between bee colonies. Varroa parasitization have been found to cause viruses in Apis mellifera colonies, such as deformed wing virus and acute bee paralysis virus complex. Varroa mites are extremely deadly to honeybee colonies. In the United States and Europe, 95 to 100% of bee colonies infested with these varroa mites were non-existent within 3 to 4 years. So this table just shows threats to honeybees all around the world. As you can see, 3 out of the 6, the varroa destructor is their number 1 threat. And here in Latin America, it is their second biggest threat. So the varroa destructor impact on an individual honeybee. They can cause decreased body weight and water content of emerging bees, deficit in sperm production and reproductive fitness due to the decreased body weight. Flying, homing, and orientation is altered in foraging bees, and their immune system is significantly weakened. And this is possibly due to the mites feeding on the fat bodies. Now, their impact on the honeybee colonies. The size of the bee colony directly correlates with the progression of infestation. With more brood and bees, mites are able to reproduce more quickly. Feral bee colonies in the United States were almost completely wiped out within 10 years of the varroa destructor entering the US. How varroa mites spread between colonies. They can spread when infested bees drift into another colony, when beekeepers add bees or brood into an infested colony without knowing, transporting colonies from one area to another, or they can spread as colonies rob honey from one another. So this map just shows the trends in honeybee population. As you can see, it's decreasing up here in this region, increasing in the United States, and it's pretty steady down in South America. This map shows the Varroa destructor infestation. So this just shows the distribution of the Varroa mites. And as you can see here in Australia, they are absent as mentioned before, but as you can tell, that is a lot of red, just showing the Varroa mites are present. How to help the bees. One method of controlling is chemical control. Several chemicals such as fluvalinate and flumethrin have been used to help control the Varroa destructor. These chemicals are found to only have killed the mites on the adult bees, but not the mites within the capped brood. Varroa mites can build resistance to these chemical substances though. For example, the Varroa mite resistance to fluvalinate is now widespread due to DNA mutations. Synthetic aericides have been shown to be helpful in preventing colony losses. If chemists and agriculturalists worked together to create a new pesticide that targeted the mites more effectively without harming the bees and contaminating bee products, colonies could thrive through an infestation. Another method of regulation is thermoregulation. Since the 1970s, hypothermia has been used to help control mite infestation. Since bees can withstand heat, heating the hive has been proven successful in controlling an infestation. Heating the hive doesn't harm the bees and is successful in regulating the infestation by mite reproduction discontinues at 36.5 degrees Celsius, and mites die at 38 degrees Celsius. There are a few hive heating devices available on the market, but then again, there are costs associated with these. Essential oils are another means of regulation. Some research has been done regarding the use of essential oils on Varroa mite infestations. Data from this experiment showed that colonies fed supplemental diets with mint oil and camphor showed the most fallen mites. And lastly, and the most interesting to me personally, was selective breeding. There has been some research looking into selecting resistant or tolerant honeybees to build resistance in the Apis mellifera species. This would provide a longer-term, more sustainable solution to Varroa destructor infestations. Geneticists look for behaviors such as recapping, hygienic behaviors, grooming, and swarming to help increase resistance at a colony level. The economic importance of controlling Varroa mites. Varroa mites have impacted every country that they reside in negatively. Varroa mites have caused billions of dollars lost due to how many colonies have been killed off. Beekeeping production costs have also increased significantly, resulting in loss of profit margins. The Varroa destructor is one of the primary reasons Apis mellifera colonies are declining all over the world. The amount of damage this parasite can do is astounding, and new means of controlling infestations is needed to prevent more colonies from dying off. Varroa mites are very evolutionized and will continue to evolve as bees evolves. There should be more urgency in protecting our colonies. Thank you.